Okay, welcome back everyone to the last session of the day, and our first speaker here is going to be Karl Broman. Karl.
Thank you. I'm going to talk about a generic model for genotype reconstruction in multi-parent populations. There's a wide variety of multi-parent populations, including recombinant inbred lines like the mouse collaborative cross and magic lines in plants, and advanced intercrosses like heterogeneous stock mice and rats and diversity outbred mice. They're all formed from some of inbred strains, and their genomes are a mosaic of those founder chromosomes. A key step in the analysis is to reconstruct that pattern along the chromosomes using SNP genotypes. Here's SNP data in a 1 megabase region for a single DO mouse, and the 8 inbred founders. Open and closed circles are the 2 SNP alleles being homozygous for one allele or the other, and the gray dots are heterozygotes. So we use all this data to calculate across the chromosome, at each position on the chromosome, the probability that that DO mouse has each of the 36 possible genotypes, the 28 heterozygotes and the 8 homozygotes. In this case, it's homozygous in the segment, heterozygous AD in the second segment. Like we can threshold those probabilities to get inferred genotypes. You could skip this step and just do a genome scan at SNPs, just like in GWAS, do a t-test at every SNP. But the genome reconstructions can, in some cases, give more powerful QTL analysis, and the reconstructed genomes are really useful for diagnostic purposes. Now, here's an example DO genome. This is what we're trying to figure out, but with dense SNP data on the mouse and its the founder strains. The main approaches to use to do this genome reconstruction are all use a hidden Markov model. You have these underlying genotypes that we're trying to figure out, but what we observe are SNP genotypes overlaid. This model is governed by 3 sets of parameters: initial transition probabilities that govern basically the genotype process along the MPP chromosome, and emission probabilities that really concern SNP genotyping error. RQTL2 has implementations of this for quite a variety of different cross types, and basically it varies in these initial and transition probabilities. I've spent quite a long time studying those MPP chromosomes, first in 8-way recombinant inbred lines, but I've spent a lot of time looking at that, calculating these transition probabilities for different cross designs, and it can be quite tedious. And one may ask, is it really necessary? So what I'm proposing today is just a generic model we could maybe apply generally to a variety of cross designs without having to work out the exact details for every cross. And basically you imagine that you have this founder population of K inbred strains in some known proportions, and you do n discrete generations of random mating with really large populations. In that case, we can calculate the transition probabilities and the initial probabilities on a fixed chromosome at generation n, we could just use these probabilities in place of the exact ones for different populations. You need to specify these founder proportions, which are probably known from the design, and you need to know the number of generations or kind of effective number of generations of random mating, which we could choose to match this map expansion, which we can calculate exactly. If you apply these sort of general model to the DO, you get really negligible differences. A genome scan, you can't really tell the difference between the 2 methods. And if you calculate the differences, the biggest ones are about 0.01. If you apply this in the collaborative cross, for autosomes, they look exactly the same. For the X chromosome, there are some important differences that really relate to the X chromosome in a collaborative cross mice only includes 5 of the 8 founders. So here for this CC038, there's a, using the more exact model, using the approximate model, there's one difference of in the approximate model, you can't distinguish is it B6 or NOD in this one region, where in the exact model, you know that B6 shouldn't be there, so it has to be NOD. But so I'm proposing this generic model for genome reconstruction in multi-parent populations. You have to specify the relative proportion of founders and this effective number of generations of random mating. The basic conclusion here is that Richard Mott's Happy software is effective, and we could have really just used it generally. The only difference here is I'm allowing that founders are not in equal proportions, which has some use in plants. There's a paper on bioRxiv. Thanks.
Brilliant. Thanks very much, Karl. Super. Any questions for Karl? Comments?
I have one.
Go on.
So Karl, what about imputation or haplotype reconstruction without knowing the founders? So I'm thinking of the CFW, Richard and Jonathan CFW paper, for example, where they had to reconstruct or to impute basically without knowing the founders. Are the more generic tools that come from human genetics usable? Do we need something specific for cases where we know the founders?
I mean, knowing the founders gives you really important information. And so if you have it, I would use it. In the case that you don't know the founders, yeah, I would talk to Richard and use his Stitch package. Or, you know, there are other similar packages that are just assuming that you're coming from some founders that you're reconstructing at the same time too.
But you know that we gain a lot by specifying the founders. We do gain a lot.
Well, yeah, of course. I mean, because you can ascribe effects to particular strains is really useful.
So
and I mean, in addition, you can use the you know, if the founders are sequenced, you can use that to fill in or, you know, basically impute at all, you know, all polymorphisms among the founder strains.
Okay.
Sorry, Abe.
I think one difference is that Stitch doesn't output founder haplotypes. It fills in SNPs. It does that kind of imputation thing that we're used to, but it doesn't output founder haplotypes. Robbie has said it could, and that he'll give an update, but so far that update doesn't exist. I was wondering, Karl, could you say something briefly about speed? Are these easy things to do when you have a lot of SNPs and a lot of individuals? And are there any advantages or disadvantages to one approach or the other in terms of just the computation?
It's not too different. It's faster than using the exact probabilities for the DO, because the exact probabilities for the DO are kind of a bear to calculate. And so you can save 25% computation time doing this. But if you think of the total time that you're spending on the analysis of a project, I think this part of it is relatively small.
Yeah.
So I don't think it may. I don't think it's I don't think it matters too much.
Perfect. That's what I needed. Thanks.
Thanks very much, Karl. Okay, great. We can move on to our next speaker, Xianmin Zhu from Shanghai Tech University. Xianmin.
Can you hear me?
Yes, we can indeed.
Okay, thanks.
Are you actually at the moment based in Shanghai?
Excuse me?
So are you actually at the moment in Shanghai, which must be, what, early morning there? Some crazy.
Oh, yes. It's actually 3 o'clock, early morning. So,
right. Sorry about that. And thanks very much for making the effort to present. Thanks.
okay, can you see it?
Yes.
Okay, let's start. So thank you very much. And today I will talk about the integrated transcriptomic analysis reveals the unique NASH progression in the leptin deficient rats. And first I will spend a little bit of time talking about the babies in our lab. You know, by taking advantage of the CRISPR-Cas9 technology, we already, you know, made several rat models, including the diabetes, hypothyroidism, and recently we also made the immunodeficient rats for the oncology study. And from this picture, I will then focus on the NASH phenotypes in the leptin deficient rats. And we know that the NASH is a complex liver disease, but it's not just, you know, a phenotypes in the liver, because it has interactions between the environment and the genetic background of patients. And as you see in this picture, it involves in different cell and tissue types. There are many models to study NASH generated by the gene mutations or the data induction, in which the leptin deficient mice are widely used as an FOD model. We know that leptin is a key regulator of food intake, glucose, lipids, metabolism, reproduction, so on and so forth. However, it should be noted that the OB mice actually did not develop a spontaneous NASH. So how about rats? Firstly, when we generated rats, we look at the phenotype. They are very similar to the mice. They are obese and also diabetic. But when we look at the phenotypes, they are a little bit different. For example, in the middle, this rat developed the kidney disease. However, this was not reported in the mice. But when we look at the close of the serum parameter, as I highlighted on the right, you can see the ALT and the AST levels increase dramatically at week 16, which implies that you know, they may have inflammation after week 16. Then we look closer by the HE staining and oral rat staining of the liver sections. On your right, you can see we highlighted that these mice really develop the NASH phenotypes such as stenosis, the lymphocyte infiltration, and also ballooning. So to address the temporal progression of NASH, we did RNA sequencing of the liver samples from 4 weeks all the way to 48 weeks. And as you can see in the figure C, the NASH progress stepwisely. For example, from the lipid metabolism, then the insulin resistant genes, and then inflammation, ROS, ER stress, to all the way 32 weeks, you can see the mitochondrial function. Very interesting, when we look into the lipid metabolism, the overall expression of the genes did not change much between the mutation and wild type. However, we observed that the liver's status actually is correlated with elevation of the gene expression of a small number of rate-limiting enzymes shown over here, and you can see the list over here. And also we observed the upregulation of the inflammation response, and also this is correlated with the activation of JNK and NF-κB pathways as shown on the right in the Western blot. We know that during the inflammation, there's polarization of the macrophages from M2 to M1. We look at the genes, for example, the M2 related genes are downregulated, but M1 are, gene is upregulated. Consistently, you can see the number of the macrophages of M1 is upregulated here, and the M2 number are decreased over here. So far, we characterized the NASH phenotypes, and then we asked is this molecular etiology of the NASH in the rats evolutionarily conserved to human beings? So we compared the differentially expressed genes of rats to the previous study of the human patients of the healthy obesity, NAFLD, and NASH. And we found that actually the rat has 42 genes that share with human beings. And in the previous study, the mouse only share like 18. And also we compared the gene expression of the drug candidates, and we found that our leptin deficient rats can evaluate the efficacy of the drug candidates. To summarize, we monitor the hepatitis gene expressions through a time course from week 4 to 48. And also we found that NASH progress in the multiple hits in a stepwise manner. And liver statuses is correlated with the elevation expression of subgroup of rate-limiting enzymes. And the NASH phenotype appears at week 16 and with a highly expression of these pathways. And this also involves the activation of NF-κB signaling pathway and immune cell infiltration. And at last, we think the leptin deficient rats are valuable animal models to screening potential biomarkers and drug candidates. With that, I will thank especially Ping Lu, who did most of the work, and all the other lab members, and also our grant agents. Thank you very much.
Thank you very much, Xianmin. Do we have any questions? Raise your hands. Amelie?
Yes, I guess I'll say that if you ever decide you're interested in, doing a cross between your rat model and some mapping population, for example, I have a friend who has developed some automatic scoring of tissue histology slides for the NASH disease. Well, he did it for humans, but I guess you could apply that to your phenotyping as well, and that would considerably speed up the process. So I could put you in touch if you were interested.
All right, thank you.
Great. Okay, if there aren't any further questions, we move on to our next speaker, which is Manuel Delpero from the Humboldt University in Berlin.
Everyone, the title of my presentation is Identification of Novel QTL linked to the Metabolic Syndrome in the Berlin Fat Mouse. The Berlin Fat Mouse is a model for obesity and the metabolic syndrome in particular. And the aim of our study was to identify genetic variants associated with traits of the metabolic syndrome using two particular BFMI lines, the 861S1 and the 861S2. These two lines are very interesting because they are genetically very similar, but despite this similarity, they differ for metabolic traits. In particular, the S1 line shows insulin resistance and also hepatic fat storage, while the S2 line is insulin sensitive and doesn't show hepatic fat storage. So with these two BFMI lines, we created an advanced intercross line population. And in particular, for our project, we focus on the A allele generation 10. This population contained in total 397 male mice that we use for phenotyping and for genotyping. In particular, we collected gonadal adipose tissue weight, liver weight, and blood glucose at 25 weeks, and we genotyped the entire population using GWAS array and CASP-assay. With our phenotypes and genotypes, therefore, we performed QTL mapping. And we identified three main regions associated with the collected metabolic traits. In particular, one region on chromosome 3 associated with gonadal adipose tissue weight and blood glucose, one on chromosome 15 associated only with gonadal adipose tissue weight, and one very huge region on chromosome 17 of around 20 megabases associated with all the three metabolic traits that we collected. So gonadal adipose tissue weight, blood glucose, and liver weight. In particular, the QTL on chromosome 17 is represented in the figure. And as we can see, the region is quite large, up to 20 megabases. So since we identified quite large regions associated with our traits, we perform a prioritization of the genes in each QTL region. For the prioritization, we use three different lines of evidence, in particular, whole genome sequencing data from the parental lines, DS1 and DS2, gene expression data from microarrays, and pathway informations. With these three different lines of evidence, we created a decision tree prioritization approach that is represented in the figure, which allowed us to score every gene in each QTL region from the most interesting one to the less interesting one. For example, applying the prioritization approach on the QTL on chromosome 17, which is the most interesting one, associated with all the 3 metabolic traits that we collected. In this region, we identified in total 231 protein coding genes. Having a look at the whole genome sequencing data from the parental strains, we identified that only 27 of these genes are polymorphic between the parental lines. Indeed, as we said before, the parental lines are genetically very similar. Afterwards, applying the prioritization approach on the 27 polymorphic genes, we identified 2 genes, plasminogen and acetyl-CoA acetyltransferase 2, to be the top candidate genes for this QTL region on chromosome 17. In particular, both PLG and ECAT2 carry missense variants in domains. PLG also shows variants in regulatory regions such as promoters, CTCF binding sites, and enhancers. And we saw that ECAT2, in addition to that, is also differentially expressed between the parental lines in the liver and in the gonadal adipose tissue. So in conclusion, we identified 3 regions associated with the metabolic trace differences between 2 closely related inbred lines, one on chromosome 3, one on chromosome 15, and one on chromosome 17, very interesting. And we observed that the prioritization of the genes in the QTL on chromosome 17 leads to PLG and ECAT2 as causal for ectopic fat deposition and the hyperglucose homeostasis that we observe in mice of DS1 line. Thank you for your attention, and if you have any further question, please ask.
Thank you very much, Manuel. That's great. Do we have any questions for Manuel? I see no hands. Manuel, perhaps you could just remind me, I might have missed this at the very beginning, the difference in the selection for these 2 lines. What were they selected for, S1 and S2? So
let's say that at the beginning, we had the founder of these 2 lines that was selected for the high body weight. And then simply they were divided for metabolic traits. Let's say that the S1 was therefore selected for insulin resistance and ectopic fat storage, while the S2 line was kind of the reference of more healthy and not showing hepatic fat storage and also insulin sensitive, let's say.
Okay, thank you very much. Great. Okay, if there aren't any further questions, we can move on to our next speaker. And that's Brittany. How do you pronounce your surname? Leger? I hope I get
Leger.
Leger, right, okay. Very good. From UCSD. Thank you.
Can you all see my slides?
Yeah.
Perfect. All right, so I'm Brittany Leger, and I'm in Abe Palmer's lab. And today I'll be presenting on our ongoing work using network co-localization in the genomic landscape to try and find shared genetic control of BMI and height between humans and rats. And I do want to mention that primarily this work has been done by Dr. Sarah Bryn Rosenthal in collaboration with Trey Eideker's lab. And we'll get to why I'm presenting it later. So the whole basis of our study is how do you synthesize human genetic data and rat genetic data? So here we have GWAS from our HS rat population for BMI and for human BMI from the Broad Institute's giant population. And if we just take the intersection of these two data sets, we get a very small number of genes. And unfortunately, we are concerned that we're losing a lot of substantial genes that are causal because they're being filtered out by limitations of how we run our studies. And so what we want to do instead is we want to try and use network propagation to find a network of regulatory genes rather than just a direct intersection. So how that works is we have our human gene set and a rat gene set that I mentioned before. And what we do is we use PCNet, which is a database of physical and functional associations among human genes. And so we take the human orthologs of our rat genes, and again with our human genes, and we make a molecular interaction network. So based off of PCNet's annotations, we calculate the proximity and network space between our seed genes and the genes that are associated with our seed genes in PCNet's interactome. After we do this propagation, we then take, we use a Brin's tool, which is called NetColloc. And what it does is it co-localizes our two networks, and then based off of a threshold, it selects for genes that we think are relevant in both networks together. And so that gives us our finalized intersecting network. And so after we have our intersecting network, then we can start doing the fun stuff, which is analyzing it and trying to figure out why it looks the way it does and what's relevant in it. So one way we can do that is we can do functional and gene enrichment. So you can do a multi-scale community detection, which basically we use the high-def algorithm, but algorithmically it makes a hierarchical structured system from our network. And so we get larger networks with smaller associated gene networks inside of them. And then when we do functional enrichment, we can take something, for instance, like the Go gene associations, and we can annotate those communities or systems to try and figure out what these functional groups are and how they actually work biologically. So we're studying this in BMI initially because it's a highly heritable trait with lots of genes. It's very polygenic, and there's a clear through line biologically between human and rat BMI. And so rather than taking our small number of intersecting genes between our two data sets, now we can have this massive network of genes. And here we show our network, where in blue is our rat BMI-C genes and red is our human BMI-C genes. And then in white are all of the genes that are significantly intersecting between these two networks. And when, as a quality control, we looked at the size of this network relative to other networks. So what we did was we took our rat BMI network and co-localized it with different human traits. And we found that the rat human BMI networks are significantly more large than the networks that we produced using other human traits, even height, which you would expect would have a strong correlation. And I won't be showing this data, but we also did this in height as well. And we found that height interacts or intersects well with itself, but not as well with BMI in the same way. So after we took our network there, we can now do our functional enrichment that I mentioned before, and we can get a BMI systems map. And we can find all of these different systems that have associated genes in our network. And so in each circle is a system, and the size of the system determines the size of the circle. So as you can see, as we get further down in a system hierarchy, they get smaller. And then in black is the fraction of rat BMI-C genes, in pink is the human BMI-C genes, and then in white is our network implicated genes. So those were not directly in either of our initial data sets. And you can see that most of our systems have all three sets. It's rare if you only have one set of C genes in there. And a few of our systems actually have previously been associated with BMI and metabolic rate. Another analysis you can do from our system is we looked at the tissue enrichment of our genes that we got out of our system. So this is, here we took our intersecting network, and we looked at BMI primarily, but we can also look at height. And we found that there was substantial enrichment for brain and neurological tissue expression of our C genes for BMI, but for height, we found no particular enrichment. And then on the left is, again, our BMI network genes and the specific tissues within the brain that are enriched. And so I hope that I convinced you that NetColoc is a really interesting tool to try and look at how rat and human genetic data can intersect, and specifically that there is a neurological role of a genetic neurological role for BMI in both of these systems. And the reason that I'm presenting this is because I'm currently working on validating our findings using a independent mouse data set, the International Mouse Phenotyping Consortium. But once we have validated the legitimacy of this tool, I'm really interested in trying to determine how we can use it to determine which models of drug abuse best correlate with human drug abuse behavior. And with that, I would like to thank, of course, Bryn again, Trey and Abe, as well as the entirety of the Palmer Lab and the IDACR Lab. And I'd be happy to take your questions.
Great, thanks very much, Brittany. Do you have any questions for Brittany?
Whoops, excellent.
I don't see one. Okay, that's brilliant. In which case, we're going to move on to our last speaker for today. That's Amy Dunn from the Jackson Laboratories again. Amy.
Yes, thank you. I have 15 minutes, right? Okay.
Yes. Sorry, I just muted myself. The answer was yes.
Okay. I was worried with all of the other 5-minute talks. Okay. Can you see my PowerPoint?
Yes.
Okay. Hi, so yeah, I'm Amy Dunn. I'm an associate research scientist at JAX. I work with Catherine Kazarowski and Kristen O'Connell. And today I'll be talking about some of the gene environment interaction studies that we've been doing in a genetically diverse mouse model of Alzheimer's disease. So Alzheimer's disease doesn't need much of an introduction, especially after Anne's introduction earlier today. But I will point out that Alzheimer's isn't just a disease of memory impairment. That's kind of the most salient trait, but it's also characterized by metabolic dysfunction and other non-cognitive symptoms. There are both familial and sporadic forms of Alzheimer's disease. Sporadic Alzheimer's disease is more common, and it's also quite heritable. But most of the heritability is unaccounted for by what we know about Alzheimer's genetics. So there's a big chunk of missing heritability, and our lab is interested in trying to chip away at that missing heritability by looking at gene environment interactions that may explain some of this missing heritability. And in addition to genetic factors, there are other factors that go into a person's individual risk for Alzheimer's disease. There are sex differences in Alzheimer's. Women make up a majority of cases, and they also experience the disease more rapidly than males do. In addition to those biological factors, modifiable factors and environmental risk factors account for another big chunk of Alzheimer's and dementia cases. But we know that environmental risk factors don't affect everyone equally. So some people are more or less vulnerable to environmental risk factors. And that's what really interests me and really drives my research, is trying to figure out what those individual differences are that will predict whether a person will be vulnerable or resilient to an environmental risk factor. So this is a challenging question to address in humans. So to overcome some of those challenges, we use a genetically diverse mouse population of Alzheimer's called the ADBXDs. And we created these by crossing the 5X-FAD model of Alzheimer's, which is a really well-characterized mouse model of Alzheimer's on a V6 background. And we cross them to BXD strains. So what we get is expression of these Alzheimer's mutations against a genetically diverse background. And these function as a really good model of the phenotypic and genotypic heterogeneity that we see in the human disease. And I'm using these mice to ask what are those genetic variants that might be contributing to a variable response to high-fat diet in the context of the disease. So to give you an idea of the scale and the timeline of this study, we have, we start feeding mice a high-fat diet or continue them on chow starting around 2 months of age. And then for the duration of their life, we take longitudinal cognitive and non-cognitive phenotyping, and we collect tissue at 3 time points: an early adulthood, a pre-symptomatic stage, and a later symptomatic stage. And in this study, we have almost 40 strains of ADBXD mice, and we shot for an N of 5 mice per strain, sex, diet, genotype, and age. And this works out to almost 5,000 mice that are a part of this study. And we first asked kind of basic questions like what is the effect of high-fat diet on metabolic and cognitive traits? In this case, we looked at weight and short-term memory. And high-fat diet had an expected effect on weight. So in the dotted lines here, you can see that high-fat diet caused excess weight gain in both females and males between 2 and 14 months of age. And that was true for our non-transgenic mice and for our Alzheimer's mice for both sexes. I will point out that our Alzheimer's mice in red weigh significantly less than their non-transgenic counterparts from these pre-symptomatic stages even. And that's something that we see in humans. So humans with Alzheimer's disease often experience weight loss or failure to maintain weight in the years preceding a diagnosis of Alzheimer's disease. In terms of short-term memory, so this is a short-term memory measurement taken from fear conditioning. Again, looking at progression of short-term memory decline between 3 and 14 months of age. In general, our Alzheimer's mice in red again performed worse than their non-transgenic counterparts, especially females. So females in our population experience earlier and more rapid decline, which again is really similar to what we see in humans. And I will point out that in the dotted lines, the mice fed a high-fat diet did not have worse cognitive function, and they didn't decline faster in any group. So, so we weren't seeing a significant main effective diet at a population level here. And that was somewhat surprising to us given some of the literature on diet and Alzheimer's disease. But of course, one of the strengths of this population is that we can look at how each individual strain responded to high-fat diet. So that's what I'm showing here. I'm showing a change in body weight between 6 and 14 months of age and a change in short-term memory function over that same time period. And you can see that there's a wide range in susceptibility of Alzheimer's mice, both females and males, on chow and high-fat diet to this Alzheimer's related weight loss. And similarly, there's a wide range in vulnerability and resilience to Alzheimer's related cognitive decline across this time. And I can kind of distill these 4 plots into one value in a measurement I'm calling a susceptibility score. So this is looking at the differential between Alzheimer's and non-transgenic mice who are on chow and high-fat diet in both weight phenotype and memory phenotypes. So mice on this side of the axis, on the left panel here, have exacerbated weight loss if they're on high-fat diet, and Alzheimer's mice on this side of the axis are able to gain or maintain weight better if they're on a high-fat diet compared to their non-transgenic counterparts. My mouse appears to be frozen, but on the right side here, the same thing. So mice on the underside of the axis mice on the underside of the axis here have exacerbated cognitive decline if they're on high-fat diet. Mice on this side of the axis have improved cognitive function if they're on high-fat diet. And I've plotted males and females here. So you can see that there's a significant sex by strain interaction where some strains, females in some strains are responding poorly to a high-fat diet, either metabolically or cognitively, where their male counterparts might be performing better on high-fat diet and vice versa. And I asked what the relationship was between the susceptibility to weight loss and susceptibility to cognitive decline. And what I saw was a fairly modest relationship where the female mice that were better able to maintain their body weight while they were on a high-fat diet were the same females that were performing better cognitively. And I don't see that same relationship in males. So this seems to be probably a sex-specific benefit that we see some female strains experiencing in response to a high-fat diet. We, of course, want to determine the genetic underlying factors leading to this differential response to a high-fat diet. So we started with sex-specific GWAS hits for human Alzheimer's disease. And there are a dozen or so sex-specific GWAS hits. And there was one, a serpin locus, that serpin B1A in mice, that seems to be significantly associated with both metabolic response and cognitive response to high-fat diet in males and females. But the relationship is going in the opposite direction in males and females. So for example, females with the D allele at this locus are less susceptible to weight loss. So they lose less weight, but they also experience greater cognitive decline. So there seems to be a disconnect between the genetic contributions to metabolic and cognitive response to high-fat diet. And that's in contrast to some other AD-associated loci, like the ApoE locus, in which case it's associated significantly with both metabolic and cognitive response to high-fat diet, but in the same direction in males and females. So in conclusion, the susceptibility to high-fat diet detrimental effects, both cognitively and metabolically, varies with sex and genetic background. Genetic control of high-fat diet response may be different in males and females. And we're working to validate this serpin locus not only as a sex-specific GWAS hit in mice, but also as a potential sex-specific mediator of high-fat diet response. And I wanted to leave off with a little plug for another lab mate who is looking at additional sex differences in this mouse panel. So Bree Gurdon is a graduate student in the lab who quantifies different cell types and pathologies throughout the brains of our entire ADBXD panel. And what she's found is that females have a much higher neuroinflammatory burden than males, which this is something we'd really like to follow up on because high-fat diet itself can induce neuroinflammation. So I'm really interested to see if the neuroinflammatory response to high-fat diet is also different in males and females and across our panel. So with that, I'd like to thank the Kazarowski and O'Connell labs. Many of the people on this list are the people who are actually doing the work in this study, and of course our funding sources. And with that, I will take questions.
That's great. Thanks very much, Amy, for that. Do we have any questions for Amy? Okay, can I ask a question, Amy? At the very beginning, you talked about setting up your cross between the Alzheimer's strain and BXD. Was that a reciprocal cross, or does it just go one way? What's the direction?
Right, they're just F1, so they were female 5X FAD mice and male BXD mice.
Right.
So the mother was always the same, and it was the father who was different.
Why this way and not the other way around?
So that we didn't see genotype effects in childbearing and mothering.
Right.
Which may differ across the BXD strain, but won't differ if we use the same females.
I guess the reason why I'm asking is because, I mean, that's one obvious factor that will play a role, but if you don't have a reciprocal cross, that you don't have necessarily equivalence between the crosses, right? So you could have imprinted gene effects and similar things where the effects might be quite different depending on which direction the cross goes. But I mean, obviously you have a very good rationale for setting up this way because you don't want the genotype effects on that.
Right.
Great. Okay. Right. If we haven't got any further questions, I don't see any hands. I don't see any people waving at me, or they just want to say goodbye. I'm not sure. So that brings us to the end of the first day. We've really had some brilliant talks. Thank you very much indeed to all of the speakers and to everyone for participating. I really enjoyed that. It's gone dark here now. But I'm sure if you're in San Diego, you still have the full day ahead of you. Martin, I see you're in Glasgow and the sun's still shining, but that must be a background. Yeah,
there's a background.
It's a background. Yeah. That's right. Okay, so thank you very much again, everyone. I shall see you tomorrow again at the same time, 1500 hours British Standard Time, and we'll have another exciting day ahead of us. Thank you very much, and I'm going to close the meeting now. Thank you, and bye-bye.